So, hello, good afternoon, everyone. I hope that all you hear me well. So first, I would like to thank the organizer for providing the opportunity to present in this wonderful conference. It's really nice to be here. And today, I would like to specifically talk about our experience on how we can use computational modeling with combination with experimental techniques to design a sustainable detergent. So I would start with a brief introduction about enzyme and the role in detergent. As you see in this figure, enzymes are used in many different industrial sectors, but there is also increased demand and interest to use enzyme in different applications. And one of the largest application, which holds about 30% of the enzyme market, it's enzyme used in household and laundry. So it's expected the market for enzyme in total to reach up to 20 billion US dollar by 2025. And 1/3 of those enzymes are used in mainly in household and laundry. But why we use enzyme in laundry? In principle, modern detergent, as you see here in this figure, are composed of several ingredients, including mainly surfactant, which are used to stabilize soil, softener or builder, which are mainly based on the polymers. And enzyme, as you see, there are different classes of enzyme are used in detergent and other ingredient. Here you see that enzyme are important part of the detergent, and several enzyme classes are used to remove different substrate. And one of the most important enzyme in most of the detergent are proteases, as you see here. So proteases are used commonly to remove protein-based stain like egg, blood, or grass from the fabric stains. And there is a high demand to use enzyme in detergent due to the sustainability, and there is also always unbroken need to identify new enzyme which are highly performing and can be used in detergent. But as we know, enzymes are biological catalysts, and they have some challenges when they are used in environments such as detergents. So they have to tolerate high temperature, very, let's say, alkaline pH, which are used in the detergent, and they have to also tolerate a lot of harsh chemical, including surfactant or oxidating agents. So this means that there is also a big bottleneck in development of novel detergent based on biological catalysts to develop always enzyme which can tolerate such a environment. And as you see here, there is also high trend also to use enzyme because we could use detergent at lower temperature to have a higher performance. But how we can design enzyme? In principle, as for non-biologists, so enzyme can be engineered using enzyme engineering techniques. In principle, they are classified into 3 categories. As you see, methods rely on directed evolution, rational design, or semi-rational design. So in directed evolution experiment, which are in principle based on natural selection. So the idea is that you have a protein, so which is encoded by amino acid sequences. You can generate a random mutational library, and then you can use a selection criteria or a high throughput screening system to identify better variant using a functional assay and then characterize them. So this relies mainly on experimental techniques, but there will be no general molecular understanding from the improvement of enzyme. So due to the importance of this method, Frances Arnold, one of the Nobel laureates in 2018, was awarded the Nobel Prize. But now, based on existence of more protein structure, so we have method rely on computational design. As you see, in such a method, we use the knowledge from the structure-function relationship of protein to identify a specific position in an amino acid chain of a protein, and then to use method of mutagenesis to identify better variants. So mainly here, so we know that there are 20 amino acids in the nature, so we could exchange one amino acid with one other amino acid to identify better enzyme in the new environment. So these are mainly rely on so-called site-directed mutagenesis. But As you see, compared to the direct evolution approaches here, we do not use the diversity offered by nature. That's why semi-rational methods are defined, which are in principle a combination of both methods. So here we identify a position using computational techniques, but we try to generate a mutation library in a specific position. And during my talk later on, you will hear this word. So we used mainly so-called site-saturated mutagenesis. This means that we could exchange one amino acid in a protein with 20 other amino acids. So there is a big trend to use such a method because we could reduce much experimental effort and to accelerate the protein engineering campaigns. So with this background, I would like to focus on the topic of today. So the project that I am presenting today was performed in so-called HiCast. HiCast stands for Henkel Innovation Campus for Advanced and Sustainable Technologies. And this project was funded by Henkel, one of the key players in enzyme, in detergent. And as you see here, the idea and the aim of HiCast was to understand, to develop more sustainable and novel detergent using molecular understanding of interaction between the enzymes, surfactant polymer in the formulation, and the material surface. So for this four different expertise of four different groups at University of Aachen, my former institution, we team up in order to understand the interactions and also to design the formulation. So as you see here, so we used method based on biotechnology or protein engineering, catalysis and synthesis, method rely on colloid and computational modeling. So today, I would like to present one of the highlights of our work. So our first investigation in HiCast shows that in one novel detergent, we could, by combination of some polyelectrolyte, as you see here, we could have much better performance of the and cleaning of a surface. So you see clearly by combination of enzyme and polymer, you have much better cleaning on the surface of the cotton. So we call this effect a boosting effect. And due to presence of the enzyme, we believe that this has been this effect is because of interaction with the enzymes. So to understand this, so we combined several methods from modeling, enzyme engineering, and biophysical characterization to understand the interactions in this complex system and also to improve it later. So as I mentioned, modern detergents are very complex. So some of them up to above 20 different ingredients. And as you see here, I show the most important ingredient that we have used. So in principle, we decided to focus on the main ingredient in the complex wash detergent. So as you see, there are surfactant, polymer, which are used in different, and for example, as surfactant, we use mainly two anionic surfactant and anionic surfactant are very important in detergent formulation. And as polymer, we use two polyelectrolyte, which one of them is a renewable resources, so-called PGA, and one which is PAA. And we showed that both of them might have effect on the boosting. As you see, so one of the third important ingredient is enzyme. So, and as I mentioned in the introduction, so the most important enzyme in the detergent are proteases. The proteases are used in order to remove the protein-based stains. And just short, what are the proteases? Proteases are enzymes which belong to the hydrolases. So in principle, they can digest polypeptide chain. So the protease that we use here, I do not go too much to the detail, but as a main structural feature, they have 5 residues, including catalytic triad and an oxygen anion hole, which they can digest a peptide in the protein. But you will hear from me in the talk that there are 2 important calcium binding sites, which are important for the structure of these proteases. So in principle, this is the proteases that we use in our studies. But to study the interaction, as I mentioned, the system is too complex. We decided first to look at the interaction between the 2 systems, and then further we try to increase the complexity of the system. So we started with a study of the protease surfactant, then we look at the protease polymer, surfactant polymer, and then all the system together with this numbering scheme. So here you see exactly how we look at the system. So let's start with our modeling. As you see, we are talking about system which is very complex, so we could not use mostly atomistic simulation, but during the project, you see when you are working with the protein, you also need to know exactly which amino acid you wanted to use for engineering. That's why we use two level of simulation, so all atom, molecular dynamic, and coarse-grained simulation. I mean, you can imagine that a system including surfactant, we have surfactant method to simulate. That's why it's much faster to use the so-called more coarse-grained model. But what is the scheme for this modeling? In principle, so we develop first all atom, and then later we used MARTINI method to scheme to derive our force field parameters for modeling this system. And what we did, we first parameterize the system using all atom simulation against our experiment, and further we develop MARTINI for larger system. For example, we use mainly MARTINI modeling and simulation. So first, we simulated every system separately. Later, for example, for binary system, we used MARTINI or all atom, depending on the complexity of system for our modeling. But later, as you see, so for our simulation, we have repeated this in two different conditions, which I come to that later. So we use simulation in the presence and absence of doubly charged ion, including a calcium 2+. So in principle, as you see in this scheme, we benchmark our system against experiment. We develop all atom. Later, we use the all atom to drive our coarse-grained modeling, and then we use this for simulation. But here you see the coarse-grained scheme. It's a standard 4-to-1 mapping mainly. As much as possible, we use the MARTINI force field. So here you see that the 3 surfactants that are used in our study, and also for polymer. So we use the same scheme in order to drive our coarse-grained model. But let's start with the first result. So as I mentioned, we observed a boosting of an enzyme and in this complex detergent. To understand the effect, first we look at the enzyme activity. As we know, protease is an enzyme which can digest a peptide, and this is the assay that we used. But as you see here, when you look at enzyme activity in the presence of surfactant, so you see that enzyme activity is lost. This is known because surfactants are mainly denaturing proteins. But interestingly, we see that when we look at the removal of stain from a surface, not in the solution, we see that we have much more solubility performance or cleaning effect in the presence of surfactant. And this effect is more than additive effect if you sum up the effect of the protease and surfactant separately. So we think that this is a synergistic solubilization from the stain of the cotton. So here I show you, so we were wondering what might be the reason. So we did the molecular dynamic. I hope I can show the movie here, but unfortunately, it's not possible, it seems. So, but we performed all atom and also a coarse-grained molecular dynamics simulation between the protease and surfactant micelle. And as you see here, so we observe that our protease is complex with 4 micelle from the surfactant. This result were further confirmed using dynamic light scattering. So, and what is interesting is that this complexation, when you look at CD spectra explain you if the protein structure because of the interaction is changed or not. So as you see, the CD spectra of this protein in the presence of the surfactant is not changed. So we do not expect any change in the structure of the protein or any, let's say, significant denaturation of the protein. So we see the boosting of enzyme in the presence of surfactant, and we see the complexation. So we believe that this might be the potential reason. So I mentioned that we did also all atom simulation, so or coarse-grained simulation of the protease and surfactant. So as you see here, so we did it for 2 surfactant, both negative charge. And what we see that there are certain residues or amino acids on the surface of the protein which are interacting mainly with a surfactant. So in order to validate this result and also to see the effect of the changes, so we identified the position which are involved in the interaction, shown here with this heat map. As you see, these are the amino acids on the surface mainly of the protein, and mainly this position are close to the calcium binding site of the protein. So we identified this position, and then further we subjected them to site saturation mutagenesis. So what does this mean? This means that we exchange every amino acid in this position with 19 other amino acid, natural amino acid, to see the effect. So as I said, these hotspots were targeted, and then we identified new enzyme variant using our assay. So here you see the result for the anionic surfactant SLES and then MES. And as you see clearly, so this is the wild type enzyme without any changes, and these are the mutation we generated. As you see here, so we have obtained enzyme variant, which they have much more performance in the presence of surfactant, and we have identified up to 190% improved if you see for some variant. But what was also interesting for us, when we use this variant and to put them in a complex function matrix, as you see here with a complex, we see also still we have identified variant which have up to 40% improvement in such a complex system. Then this shows that we have identified the position which are involved in the interaction, and we have also improved the protease performance in the presence of this surfactant. So then now let's look at the second system. So as I mentioned, the second system is protease polymer. So we titrated our system. So we, as I said, we had the protease and surfactant, and then we tried to increase the concentration of one of the polymer. So as you see here, for 2 surfactant, we see that in the presence of polymer, we have much more cleanness on the cotton. And interestingly, when we look at different assays, so we can use a very simple peptide, which will not be the representative of the final application in laundry, a soluble assay, and an assay which is much more representative in the presence of the cotton. So this means that is a protein stain which can be removed by proteases in real laundry. So you see that the effect of the enzyme and the polymer are not additive, but also here, also we see some sort of synergistic effect. So we have much more performance in the presence of both systems. So, and you see here, in the presence of this, we have up to 200% increase of protease performance, for example, for a complex protein. So to understand why protease and polymer behave like this, we perform several experimental techniques. I will not go to the detail, but in principle, for example, we use so-called fluorescence correlation spectroscopy. So in this method, you can look at diffusion coefficient of for example, the enzyme, which is labeled. And as you see here, so we see that in the presence of the polymer, we have increased hydrodynamic radius. So this means that the protease and polymer are complex to each other. And of course, to understand the thermodynamic behind it, we performed so-called isothermal titration calorimetry. And what do you see? So in principle, as you see in this model, we see that after complexation, so we have the desolvation of the polymer. And this process is in principle accompanied by heat release. It's an entalpic interaction, and the binding of these 2 moiety together is controlled by electrostatic force and hydrogen bond between the protein and the polymers. So further to validate this result, we also perform molecular dynamic in all atom and coarse-grained simulation. And interestingly, you see here for both polymer, we see that both polymer are also interacting with the surface of the protein, more or less in a similar mode of binding, close to these 2 calcium binding site of this enzyme. And so to validate this result, we also identified some position, as you see here, near to this calcium binding site, and we again subjected them to so-called site saturation mutagenesis. So this means that we exchange this amino acid with 19 other amino acid So, and interestingly, what do you see here is that comparison of the generated variant compared to the wild type. And as you see here, so we see that by mutation, we also identified variants which have higher performance in the presence of PAA or surfactant, and we could improve it up to 300%. So interestingly, this effect is present for both use polymer or polyelectrolyte. The effect is stronger, for example, for PAA, but there are a lot of optimization also possible in terms of size of the polymer and charge, and I'm not going to that details. But as you see here, so we improve this, and then later we see that we have also the same, even to improve this in the presence of also very complex detergent. So up to 60% increase of performance we have identified when we use this variant in a complex wash detergent. So the next system is surfactant and polymer as the main ingredient of detergent. So we look at effect of interaction between surfactant and polymer in two different conditions of the water. As you know, water might have some ions, which we call it water hardness or softness. And here we use two different degree of water hardness, 15 degree and 0 degree. And as you see here, it's very interesting to see that the effect of the cleanness of these two systems is much more when we use a hard water. This is opposite to the general common knowledge that you can wash, for example, or washing is better in soft water. But interestingly, what we see when we look at our solubilization efficiency, as you see here, we look at the surfactant concentration and the cleanness. So we see that cleanness is mainly possible when we have the surfactant micelle, so above their critical micelle concentration. And what is very interesting is that when we add the PAA, so we see that this effect of improvement of solubility is much pronounced when we have hard water. And this is, as you see here, so we do not have any change in the presence of the polymer. So this was very surprising us because in this system, so we have a negatively charged surfactant and negatively charged polymer. So we do not expect that this similar charge moiety they could interact, but based on our molecular simulation, we see that, of course, this is the case. So when you have a 0 degree water hardness, we do not see any interaction between the surfactant and polymer, but we see a complexation of surfactant and polymer in the presence of this hard water. And here you see the distance between these two moiety, and this result further confirmed by using dynamic light scattering. So based on this, we propose that we also have a kind of unanticipated electrostatic attractive interaction between two similarly charged surfactant and polymer, and they form some sort of nanoreactor. This nanoreactor might be responsible for higher solubility and increase of also the efficiency. So now, after studying every single system, so we look at also 3 systems. So for that, you see here the result of cleaning. As you see here, so when we have the polymer or surfactant alone, we have some cleaning, but when we use the combination, for example, of these two with high water hardness, we have improved solubility, but the effect in the presence of the protease is really improved, and we see a synergistic effect of this ingredient together. And to understand this, so this, as you see here, so we performed also a coarse-grained simulation of all the system, and surprisingly, what we see is that when we do not have any ion in our system, we do not see the assembly of this system. But when we combine this in the presence of the calcium, so we see that we have a high assembly of these 3 systems together. And as you see in this model, so we see that surfactant and polymer, before they can interact through bridging through calcium, and they can interact with the calcium binding site of the proteases. But this will not be the case when especially we do not see much clustering of those in our coarse-grained simulation. So now let's look at all the system together. So based on this combined experimental and modeling studies, so we provided a model for detergent, as you see here. So in this system, so we have interaction between the polymer and surfactant through calcium bridging, and we see also in our simulation that this negative charge, they can also interact with the stain on a cotton, so-called CO3 stain, and we see that also protease and polymer, as we said, and surfactant, they compete in interaction with the calcium binding site of the proteases. So based on this, that brings me to the lesson that we learned from this, as you see. So we could increase the performance and sustainability. So here I do not show the number, but we could increase up to 250. So this is a very important progress in the development of so-called condensed formulation of detergent. And I hope that I was able to convince you that enzyme engineering is a robust and rapidly advanced technology to tailor enzyme, even for very complex and harsh formulation. So so it was very also for us surprising to see that we could combine rational design, direct evolution, and also several experimental biophysical techniques to understand the interactions between such a complex system, including proteases, surfactants, and polymers. And in the end, we also generated some hypotheses regarding this large assembly of protease, surfactant, polymer, which they had high and superior solubility performance. And with that, I would like to thank all my colleagues. So this work was done at my former institution, RWTH Aachen University, and in cooperation with the Henkel R&D department. And I would like to thank you all for attention. I would happy to take questions.
Thank you very much for such an interesting talk. We already have some questions in the chat that I'll read out. So the first question is from Stefan. Considering the interaction spots between the protease and micelles, can the protein be considered as a patchy colloid? And if so, is this protein-protein aggregation a relevant factor for detergent performance?
I mean, we do not expect aggregation, and by that means, so what was very interesting or surprising for us was that we see every protease is complex with 4 surfactant micelle, which is confirmed mainly by dynamic and static light scattering, and we believe that this assembly together improve the exposure to the surface of the cotton, which they improve but we do not have a large aggregation of many, let's say, surfactant, and we do not observe any denaturation of the enzyme.
Thanks. And we have a second question from Rax. What tools do you use to generate the parameters for MARTINI force field, and how do you evaluate the correctness for the mesoscale force field?
Okay, so I think I was maybe I misunderstood. I was confusing, I guess. So we didn't develop any mesoscale technique. So we develop all atom model. We also develop MARTINI, and we use a standard MARTINI force field, but for proteins. This is a standard Amber, this standard MARTINI, but for the others, we drive the parameter based on the MARTINI, let's say, Bible, like the table that you have, the 4-to-1, and try to find the best or fitting bead, perform the simulation, compare it with the all atom, and also compare with the experimental data. So this was our approach to do it.
Thank you. And we've just had one final question from KMBA. Have the complexes with micelles been seen with other enzyme classes, such as lipases?
We have observed this only for 2 enzyme classes, mainly the one that have such a calcium binding site, but not for lipase. They do not share the same structure feature. So we mainly see for protease and the one that they have a calcium binding site.
Okay. Thank you very much. And they say thank you as well.